Did you hit record?
Mm-hmm.
And just so you know, we're now recording. So, uh, yeah, just be aware of that. And I also wanted to address one issue on the quiz that you guys just took. There was one question about epinephrine passing the blood-brain barrier, and in the answer, it was just missing the word "not," which is pretty important. I have corrected that, and I believe have given everybody credit if you did miss that question. But let me know if I made an error there. Now, I am going to turn this over to Dr. Hamilton, who is, in addition to being my wife, an expert in this area. She's done research in this area, and has taught this class multiple times, knows this material very well, and also was interested in discussing or presenting this particular lecture. Uh, uh, Also, I'll point out that she is sort of the teaching czar at the University of Colorado Denver. She doesn't like that term, but that descriptor. But I think it's the most succinct way, unless I have to say the director of the
Yeah,
center.
can you help me get Harry? I'm in class, dude. You are not. Can you get your...
Oh, what was that?
Somebody's there.
Sorry?
It was just somebody's noise.
Oh, yeah. No, so yeah. So anyway, she is an excellent instructor that teaches everybody how to be a better instructor, and an expert in this area. So I am handing it over to her.
Hi, everybody. I'm Dr. Hamilton, and I've taught hormones usually for the past 6-7 years now. I'm not this semester, so Dr. Olson is instead. But I am going to be going over the chapter on sex determination and sexual differentiation. This is fairly complex information, but it follows a pretty clear pathway. So we're going to walk through it a couple times and we'll spend a few days on this. I am happy to take any and all questions. Please interrupt as I'm going through. You can type in the chat or you can just unmute yourself and ask away as we go through. All right. And I recognize a few names in the class. So if I've had you before, if I've had you for Biobasis or anything, it's nice to see you again. All right. So this is chapter 3 in your textbook. But usually when I start introducing this topic, I like to think about how we're going to apply this information. So this is Stella Walsh. You can see the photo here. Stella Walsh, we are recording. It is being recorded as of right now. So it will be posted as well. So Stella Walsh was an athlete. She was an Olympic athlete. She won the gold medal in the women's 100-meter dash in the 32 Olympics, and then she won the silver in the 36 Olympics. At the time, she underwent what they called a sex test and passed. Not a lot of detail on what that entailed. Stella was actually murdered by a robber in 1980, well after retirement, and upon autopsy, showed that there was male reproductive organs. And so some people have actually called for Stella's name to be removed from the Olympic records because they claim that she is not female. No one would have ever known this if she hadn't been murdered and there had been an autopsy. There is a fascinating article on this. It's a long reads. If you just type in long reads into Google and then Stella Walsh or intersex Olympic champion, you can learn a lot more about her. But let's jump ahead a little bit. This is Santhi. She won the silver medal in the women's 800-meter race in the Asian Games in 2006. She was subjected to a sex test, and it was determined that, quote, she does not possess the sexual characteristics of a woman, and she was stripped of her medal. Then that brings us to Caster Semenya. Caster won the silver medal in the women's Castor won the 800 meter and the 1500 meter races at the African Junior Championships in 2009. She earned national championship records. She underwent a gender test. So note this time they're changing the name. It's going from a sex test to a gender test. Was not allowed to compete in 2010. Was allowed to compete in the 2012 Olympics and won a silver medal in the 800 meter race. So Semenya was actually outed as intersex and was subjected to kind of this media frenzy and coverage that tended to ignore the fact that most top athletes are utterly abnormal in terms of their body proportions or physicality, as it normally goes. Think of, for example, Michael Phelps. He has an elongated torso beyond most people's body proportions, and that is abnormal outside the bounds of what we would consider normal human physiology, and yet he is celebrated for it. Semenya was raised as a girl. She has no ovaries or a uterus, but has external female genitalia, also has undescended testes. So the impression the media puts forth is that she's enjoying kind of competitive fruits of the testosterone, having extra testosterone, but it's not so simple, as we're going to get into. So I start with these cases about some intersex athletes because this is one place where sex and gender come into play a lot. And in fact, you'll be writing a paper on this for Dr. Olson about how we kind of live in this binary world where we have to determine if someone is male or female for things like this, competitive sports. And it's not going to be a nice, easy, neat box to put everything in. So what is sex? What is it that makes someone male or female? I'm going to argue we have 5 levels of sex. And at each level, we could consider male or female. So we're going to have chromosomal sex. This is your genes, your chromosomes. Gonadal sex, which gonads are present, ovaries versus testes. Hormonal sex, what hormones are being produced by the gonads. Morphological sex, this is kind of like differences in body type. And then we're going to have behavioral sex, which is more male typical and female typical behaviors. So it's not a simple, straightforward, someone is male or someone is female. We have to look at it at multiple levels. So here's another way of kind of looking at things. We have egg meeting sperm. That's going to cause the genetic sex, right? The chromosomes. Chromosomes should give rise to the gonadal sex, which should then give rise to the hormonal sex and the phenotypic sex, changing the external features like the external genitalia, secondary sex characteristics, and then also internal genitalia. And then in the central nervous system, the hormonal secretion, the rhythms that are secreted, and also sexual behavior, male or female. So we're going to be looking at kind of sex at all these different levels. So most organisms have 2 sexes. Sexes are simply going to be defined as mutual incompatibility between the same mating type. Okay. So typically there's production of large and small gametes, and the female gametes are typically larger, they're immobile, they require more resources, the eggs. The male gametes are sperm. These are more numerous, they're smaller, they're mobile. There's different types of organisms and how sex works. We have some species that are actually hermaphrodites. They have both sexes present, snails, earthworms, most plants. There's also separate sexes, dioecious species. That's going to be most of the animal kingdom, and also some plants. Holly plants, for example, actually are separate sexes. And so if you've ever planted one and you want it to bloom the nice, pretty red holly berries that you see at holiday times, you actually have to plant both a male and a female plant. Otherwise, you will not get that. So In these slides, I've put a star at the top of really important ones. Okay, so these are kind of your cue for this is a big thing. And does everyone have the worksheet that I think Dr. Olson sent out yesterday? It's just for you to follow along. It's kind of like a skeleton note-taking sheet, but it'll be really helpful, I think, in organizing your notes. So you should have that available. All right, so the big thing we need to just talk about is the difference between sex determination and sex differentiation, right? This title, the chapter is called Sexual Differentiation and Determination. What is what? Sex determination is the initial event. This is the event that determines whether or not the gonads are going to develop as testes or ovaries. It's a one-time event. Sexual differentiation is all the other steps along the way, leading to all the different male or female phenotypes. So we can think of differentiation as this pathway, this process, lots of steps involved in it. Determination is really going to come down to one step. Okay. All right. So let's think about sexual determination. It's a mechanism. It's the developmental decision. It is occurring during embryogenesis. So this is before people are born. This decision point is being made. Aristotle wrote in 335 BCE, sex is determined by the heat of the male partner during intercourse. This is not true. We know this now, but at the time, that is what was believed. And in fact, up until very recently, it was believed that environment, and in particular temperature and nutrients, were the most important thing for determining whether or not there's going to be a male or female baby, in a sense. So they talked about factors that favor the storage of energy and nutrients, might predispose one to have female offspring, and factors that favor the utilization of energy and nutrients might influence to have male offspring. This may be true in other species. It does not seem to be true in most mammals. So when we think about sex determination, we are basically only working in the last 100 years or so. So at about 1900, we kind of rediscovered Gregor Mendel's work on, he was the pea plants and the genes and figuring out how things might work. And then in the early 1900s, we continued to kind of rediscover work around this, and we found the sex chromosomes. And we established a correlation, in fact, only in insects, between the female sex and the XX chromosomes and the male sex and XY or XO chromosomes. So this led to the idea that there's a specific nuclear component, and that is what is directing the sexual phenotype, that it's from these chromosomes. And evidence has been accumulating ever since the 1900s, really, that sex determination occurs because of this genetic inheritance, not environmental influence. Today, what do we think about? We think about both environmental and internal mechanisms, and they both can be operating for sexual determination in different species. So think of it this way, there's some master switch in our genes that turns on one pathway and turns off another pathway. We're going to talk about what that switch is. Before I go into that, I just wanted to share this because sex determination, we're going to be mainly focusing on how it works in mammals and in particular in humans, but it works differently in different species. There is no one absolute way of sex determination and differentiation. This is what male and this is what female is. So environmental sex determination is actually where after fertilization, the environmental factors like the temperature, population size will determine the sex of the offspring. There's also genetic sex determination, which is what we're going to be talking about in mammals and in humans, and that's based on the fertilization of the genes, combination of the genes. But just so you know, there is these other ways of doing things. Environmental sex determination is a whole fascinating world. It can be things like chance. For example, there's a marine worm where females are large and they attach to rocks. Males are small and they kind of float around. And if they happen to land on a female, they become male. If they land somewhere else on the ocean floor, they become female. So pure chance. There's also social environmental sex determination. So this is in a lot of coral reef fish, actually. They'll start out as one sex and then change into another depending on if there's a wipeout of the population of male or females. So the infamous example here is if you've ever seen the movie Finding Nemo, at the beginning of the movie, most of the eggs are eaten and all that's left is one little clownfish egg and Nemo's father. And really in that circumstance, Nemo would have become female because there was no female in the environment, in the social environment. So Disney ignored that fact. And here we are. Okay. Temperature is a big one. This is in many reptiles where the sex is actually determined by the incubation temperature at a key point in development. And so that can change. You can see here that many turtles, when it's colder out, they will all become male. When it's warmer, they all became female. Lizards, alligators, it's actually inverted from that. And then a lot of snapping turtles and crocodiles, it's actually this U-shaped curve where at low temperatures and high temperatures, more likely to be female, and that in between, more likely to be male. Okay. All this being said, this is not how it works in mammals, and this is not how it works in humans. So we're going to move on from that to genetic sex determination. The reason I share all that with you is because I want you to be aware that just because this is how it's done in one species is not how it's always done in other species. Doesn't mean there's one true way of being male or female. All right, so genotypic sex determination. In humans, as well as most other organisms, the sex ratio is always going to be pretty close to 50-50, not dependent on the environment. About half will become male, half will become female. In mammals, individuals are going to be heterogametic or homogametic. Hetero meaning two different types of producing two different types of gametes, the X or the Y. Homogametic, human females, we're only producing one type of egg, and that's going to be X. Okay, so fertilization of the X chromosome egg by an X sperm is going to yield female. Fertilization with a Y sperm is going to be male. This is true in humans. I just want to point out other species, it works differently in the sense that birds are actually reversed, and it's the females that make different types of gametes and the males only make one type. So it's not always working the same way in every species. And in fact, in other species, it's not even the type of sex chromosome, it's the number of sex chromosomes. So C. elegans are actually hermaphroditic, and they all have these XXA eggs, and the males actually have sperm with only one X chromosome. And then there's other species, lots of insects, where they don't have distinct sex chromosomes, but rather they have a gene that codes sex determination, and it's all very complicated. The point of this is genotypic sex determination systems are really diverse in the world out there. So again, as we're going to be focusing on humans and mammals, especially, it can be very tempting to be like, this is how things are done. This is how it always is. This is male. This is female. This is true. This is false. And really, there's a lot of gray area here. Okay. So this is Alfred Jost. He is kind of given credit for our current understanding of mammalian sexual determination and differentiation. So what Alfred did is started removing the indifferent gonads, indifferent meaning have not distinguished themselves. So at various points after fertilization, until the differentiation starts to occur, the gonads are the same for male and female. That's indifferent, hasn't been distinguished yet. So he removed the indifferent gonads from rabbits in utero and let them grow up, see what happens, and found out that all the rabbits then became female. So if he removed the indifferent gonads, they all became female. So Alfred's conclusion here is that the testes must be producing some substance required for male embryos to develop and become male. So this led to what is known as Jost's rules of sex determination. First rule of sex determination, we don't talk about sex determination. Those are the jokes, people. That's all I got. That's my Fight Club reference. All right, first rule of sex determination is the development of the gonads into testes or ovaries is going to what's determining the sexual differentiation of that embryo. Okay, that's the first rule. First rule is the gonads becoming a testes or an ovary is determining sexual differentiation. Second rule of sex determination. You don't talk about sex determination. Yes, thank you, Justin. You're with me. Second rule of sex determination, the Y chromosome is what carries that sex determining information in the mammal. There's another kind of unsaid rule here. He removed the indifferent gonads in the rabbits and they all became female. What do you think? What might be this unsaid rule? You can type it in the chat, yell it out, take a wild guess. Yeah, some of you have looked ahead on the worksheet. Females are the default, right? If we remove the gonad, they still became female. So in the mammalian world, female is the default. I'm going to say that again. Female is the default. So as we start going through the worksheet, Eric, where's the worksheet?
Send it to them as an announcement.
He sent it as an announcement, he says. So you should be able to access it that way.
Is it the study guide announcement?
Yeah.
Yes, he
Okay,
says. Okay, so female is the default. That means as we start going through the sexual differentiation pathway, and we're going to look at so many different steps in there, if at any point we go off the path, female will default from there. Okay. All right. So Jost kind of talked about 3 levels of sexual dimorphism, differences where we can be different male, female. I'm adding in those 2 other categories, right? And so you can see on the side there. So we're going to have our genetic sex or chromosomal sex. This is, do you have XY chromosomes or do you have XX chromosomes? Then we're going to have gonadal sex, testes or ovaries, based on Jost's rule of sexual determination, differentiation. The development of the testes or the ovaries is what's going to trigger the whole pathway. Then we have hormonal sex. This is what hormones are being produced by those gonads. Morphological sex leads straight from that. This is similar to phenotypic sex here in Jost's language. So the external genitalia, what does that look like? Is it male or female? Secondary sex characteristics, male or female? And then finally, behavioral sex, male typical, female typical behaviors. All right. So Jost's second rule was that it's on the Y chromosome. There's something happening there and that's triggering everything. So we're going to go through a little bit of how we figured this out. What causes males and females to develop differently? So we had this thing that we called, for lack of a better word, testes determining factor, or TDF. We called it this because we didn't know what it was, but we knew there was something that was determining coding from the Y chromosome into developing testes. We didn't know what it was or what it was doing. We knew that TDF, testes determining factor, was acting in the embryo around the 6th week of human development to start differentiating the gonad. So up until this point, we have an undifferentiated gonad. So it's going to look the same, male, female. It's all kind of identical at this point. So TDF is acting on this undifferentiated gonad at around the 6th week in human development. Does anybody know when most women determine they're pregnant? How many weeks?
The 6th.
Anywhere from like 6 to 10 even, really. The point of this is that most women don't even find out that they're pregnant until this has already started. So this is happening pretty early on in development, okay? So here's our indifferentiated gonad, okay? Germinal ridge here, don't really have to worry about it. At some point, this indifferentiated gonad is going to turn into the ovary or the testes. And this is where that TDF is acting. So TDF is acting on the support cells. So what are support cells? There's different places it could be acting. Support cells are the cells in development that are going to become the Sertoli cells in the testes or the follicle cells of the ovaries. So these are the ones that are going to be actually producing the gametes. The follicle cells are going to produce the eggs. Sertoli cells, we're going to end up supporting and producing sperm. There's also steroidal cells in that indifferentiated gonad that are going to start producing the gonadal hormones. In males, these are Leydig cells. In females, these are thecal cells. There's also connective tissue cells that are just there for the general framework. So the initial change in the gonad is a changing from the support cells. So TDF is not acting directly on these germ cells. It's acting on the support cells to start creating the proper environment to differentiate into sperm or eggs. So it's all about setting the right ambiance, setting the right environment for male or female. We also know that TDF, testes determining factor, is only expressed in cells with the Y chromosome. It's going to be expressed and change those support cells to become Sertoli cells and start to form testes. TDF is not expressed in cells that only have an X chromosome. So therefore, the support cells will default to female, default to follicle cells, and start producing eggs. So really, it's these support cells that rule sex. So at every step of the way here, we're going to be talking about some additional step has to occur to continue down the male pathway. If at any point the extra step does not occur, we're going to default female. So if we don't have TDF acting, we are not going to become Sertoli cells and form testes. We will default then to follicle cells and form ovaries. So scientists really interested in figuring out what the heck is going on here. What is this TDF? We know it's on the Y chromosome, but what is going on? So we started studying individuals whose sex did not match their genotype. So sex reversed males have 2 X chromosomes. Sex reversed females have an X and a Y chromosome. And basically, if you just remember some basic biology, during the process, when the gametes are, when we're forming meiosis and we're forming the gametes, there's crossover events of the genes, and some of the genes jump around. So there's a rare crossover event that can occur where, between the X and the Y chromosome, moving the gene that has the TDF over to the X chromosome. Okay. So if that happens, then it's possible to have an X chromosome with the TDF, the testes determining factor, and have a Y chromosome with no TDF. So this allows us to start doing some testing and really focus on what exactly is the TDF gene. So it turns out the TDF gene is the SRY gene. SRY stands for sex determining region of the Y chromosome. They named it appropriately. It probably didn't even need a name change, but we had been calling it TDF just because we didn't know what it was, right? So they were able to do genetic mapping and find out that this is a gene, it's on the Y chromosome typically, and it's called the SRY gene, sex determining region of the Y chromosome. Okay. And turns out this is gene on the Y chromosome has been around for a long time. You can see birds and reptiles do not have the SRY gene. They have a whole different sexual determination system. But platypus doesn't have it. But after the platypus, pretty much every other mammal at that point on does. Okay, so we can trace this evolutionarily. So now, because we're able to do this, we can do some cool genetic crossing in animals. So in mice, what we could do is remove the SRY gene and insert it onto just a regular old chromosome. So this actually was a big breakthrough. So transfection of the SRY sequence in the mouse can transform an XX embryo to male, and an SRY gene is necessary and sufficient to transform into male. So basically what we had here was we did some crossing and we could get female mice that were XX typical, XY minus, meaning they don't have the SRY gene. So they have the Y chromosome, but they don't have that one gene, so they develop as female. Then we have the XY SRY gene mice, so typical male. And then we have the XX with the SRY gene inserted in there, and they develop as male. So despite not having a Y chromosome. So this allows us to ask a lot of interesting questions. So they were able to do all these studies, and they essentially proved that the mouse SRY gene is sufficient for testes formation. If you just have that gene, no matter what chromosome it's on, we'll get it. Okay, so they worked and they developed this whole mouse line of this. Does an XY SRY have developmental issues like some others? So sometimes. So when you do this work with the mice and you're creating these transgenic strains, sometimes the offspring just don't come out normal, and that is true. So we do see that. I have actually worked with these mice, this strain of mice, and what I would say is that I think they're actually a little bit larger and they have a little bit of a more laid back personality, really. But they do show typical behavior, male and female behavior. So actually, like I mentioned, I did do some work with them. I was looking at brain stimulation reward, where we electrically stimulate part of the lateral hypothalamus. And in these sex reversed animals. And what we found, in case you're curious, there was no difference in basic brain stimulation reward. Like they both found equally things rewarding, but drugs like amphetamine and other stimulants potentiated, kind of increased that brain stimulation reward in mice that had the XY genotype, regardless of SRY expression. So this opens up a lot of questions about how genetic sex or hormonal differences are interacting and whether there's differences in biology in terms of motivational function. We know there's large differences in between the sexes in terms of addiction and how things work. And so a lot of times it's very easy to say this is attributed to hormones or to, X or Y gene chromosomes. And here we're saying there's some other things going on, differences on the Y chromosome to the X chromosome, regardless of the hormonal development that occurs later. Okay. So genetic sex is what's going to determine the gonadal sex, right? So we have the SRY gene and it's typically on the Y chromosome. So that will develop the indifferent gonads into a testes. If the SRY gene is not present, if it is absent, if you are XX, the gonad will develop as an ovary. So let's say you have a situation where there's a chromosomal error during fertilization and there's XXY. What do you think that person would develop as? Would the gonads develop as a testes or an ovary? XXY.
Ovary.
Female?
I'm sorry?
Female.
female?
Ovary.
Ovary?
I want to hear from a few people before XXY. All right, 2 votes for ovaries, 1 vote for testes, another vote for testes. Testes with a question mark, not sure. It's okay not to be sure. Both. So just based on the presence of the SRY gene, which is there on the 1Y chromosome, we would start to develop that testes into a testes, that indifferentiated gonad. Okay. We're going to keep talking about those scenarios and we'll get into where things also, I want to say go awry, but verge off the typical developmental path as we go on. But basically, if the Y chromosome is there, if the SRY gene is there, we're going to start to develop into a testes. I have no idea what number on the worksheet I'm on. But at the end, we'll pause and make sure that we can answer all the questions on the worksheet. Okay. All right. The rest of sexual differentiation, the rest of that process will be driven not by the sex chromosomes directly, but by the hormones secreted by the gonads. So determination, what is the chromosome? Is that SRY gene there? Are we going to develop into a testes or an ovary? That is determination. Everything else now that we're going to talk about is differentiation. It's part of that process, the pathway. Okay. Hormonal control of sex differentiation. And again, this is all in mammals. So primary sex differentiation is that determination point controlled by the SRY gene. If the SRY gene is there, we are going to develop testes. Lack of an SRY gene, or no Y chromosome, we're going to develop ovaries as the default. Secondary sex differentiation is everything else that starts to happen with the hormones, beginning in embryogenesis, so if we're still at the fetus stage, and progressing all the way through adulthood. So this is going to include everything from sex characteristics, external genitalia, secondary sex characteristics like breasts, body hair, body build, and then also behavioral. So the important thing to note is that most of our sex hormones are steroids. Steroids are lipids, right? So we're going to think about what that means from a mechanism standpoint. These sex hormones are actually derived from cholesterol. So we can see here we have cholesterol. It is converted into progesterone. Progesterone is can be converted into different things, but on the sex hormone pathway, it can be converted into testosterone. Then testosterone can be converted by an enzyme into estradiol, estrogen, or a different enzyme can convert it to DHT, dihydrotestosterone. So we typically think of female sex hormones as being progesterone and estradiol, and we think of male hormones being the androgens, testosterone and DHT. I want to caution us from thinking about them totally exclusively, because we all, male, female, we all have both. We're all going to have androgens. We're all going to have some level of estrogen and progesterone. Okay. All right. So our gonadal hormones are going to determine that phenotypic sex. So kind of what we look like in the development. So newly developed ovaries really don't secrete a lot of hormone prenatally. Prenatally, they're just not really doing anything. We're just going to continue down the default pathway. However, newly developed testes will start secreting several hormones to then direct the body into developing into a more masculine fashion. Okay. So again, male, additional steps. Female, default, we don't have to do anything. We're going to develop female. So I want us to think about this as two distinct processes. We have this kind of spectrum on this chart here. We have masculinization and demasculinization. Masculinization is going to be the induction, producing these male traits. Demasculinization is the removal of the potential for male traits. Then we have feminization and defeminization. And note that it's on a distinct kind of axis here. These are two different processes that are happening during development. And we can be kind of at different ends of this pathway here, this spectrum. We could be up here towards more masculinized. We can be down here towards demasculinized. And then same thing on the feminization scale. So what we typically think about as female is the feminization of many traits and also the demasculinization. And we think about male as being the masculinization of various organs and traits and the defeminization. But we can have things that are happening in these other quadrants. We can have parts of this pathway where we're both feminizing and masculinizing. We can have part of the pathway where we're defeminizing and demasculinizing. So what is that going to look like? Okay. So as we think about all these next steps, think about that we have to be doing both things simultaneously, and that we can be at any part of this spectrum for either axis. All right. Here we have the undifferentiated genitalia. This is about 6 weeks of age. Okay. This is the at birth male and female genitalia. The default is female. Let's highlight that again. The default is female. If we have the absence of hormonal influence, if we don't have that testy producing certain hormones, or if we have a scenario where the hormones are being produced by this new testy, but the body is not capable of responding to them, perhaps no receptors, no receptor, no effect, we're then going to default and develop female. If the androgens are present during this peak developmental time period, then we will develop male. Okay. So there's a single kind of primary organ in the embryo that's going to develop into the external genitalia. So developing one type of genitalia precludes the development of the others. So we can think again about the spectrum here. We have the masculine genital development and the female, feminine genital development. And you can't really develop both at the same time. Babies that are born not on either end of these spectrums, but somewhere in this middle portion, 1 in 1,500. That's a pretty high number. 1 in 1,500 babies are born somewhere on this continuum, not on the extreme feminine or the extreme masculine. There are a lot more intersex people than we typically think about. All right. So we have done sexual determination. The chromosomes were Y. We have the Y chromosome with the SRY gene, developing those support cells into developing a testy. Now that new testy is going to start releasing 2 testicular hormones that are going to drive masculine development. The 2 hormones are testosterone and anti-Mullerian hormone, AMH. Okay. So anti-Mullerian hormone is anti-Mullerian. It's going to cause the Mullerian ducts, which are part of this indifferentiated gonad system, to regress, to go away. We are defeminizing. Ovaries don't secrete anti-Mullerian hormone, so therefore the default is female. The Mullerian ducts will continue to develop into the fallopian tubes, the uterus, the cervix, the inner vagina, all the typical feminization of the genitalia. We also have testosterone. Testosterone is going to virilize, masculinize the Wolffian ducts to form the male genitalia, vas deferens, the seminal vesicles. So the 2 hormones are working distinctly. The anti-Mullerian hormone is working to defeminize the genitalia. Testosterone is working to masculinize the genitalia. It's also going to direct the masculine development of the genital skin into the penis and the scrotum. Okay, so 2 different hormones doing 2 different things. So here's kind of what that looks like. Testosterone is going to be synthesized by the Leydig cells in the testes. Estrogens are produced by the thecal cells in the ovaries. I want us to note that sex hormones are not solely produced in the gonads. The adrenals also synthesize a very low amount of sex hormone as well. Okay, this is going to be important later. So here's kind of our pathway we've talked about so far. We have the sex determining region of the Y chromosome. It's in the embryonic germ cells on the Y chromosome. It's going to have that testes determining protein, causes the undifferentiated gonad to develop into a testy. The testy is then going to develop into these Leydig cells and the Sertoli cells. The Leydig cells will secrete testosterone, which will masculinize the Wolffian ducts and develop direction for creating the penis and the scrotum. We have the Sertoli cells start producing anti-Mullerian hormone to defeminize and make the Mullerian ducts regress, disappear. Okay. How are we doing? Following the flow chart so far?
Yes, ma'am.
All right, I'm just going to pause for a second. I don't want to add to our flow chart till we make sure we understand this part. All right.
I have a question about the anti-Mullerian hormone.
Yes.
Is it something that's attached to the X or the Y chromosome? I guess it would have to be.
Well, so the Y chromosome has the SRY gene on it.
Right.
That's going to make the gonad become testes. When it starts creating the environment for the testes, it creates these cells, which will start producing that hormone.
Okay.
Does that track?
Yeah, I think so.
Okay. Yeah, never be afraid. Interrupt me at any point. Okay. So what would happen if we have an XY individual that doesn't produce anti-Mullerian hormone, but they do produce testosterone? What's going to happen now?
Since that SRY gene's present, they would have to develop testes. Is that correct?
Yep. So we would have the development of testes. What would the genitalia look like? Would it look like male or female? It
You
would
said
probably look like a combo. Yeah.
AMH.
Yeah. So if we didn't have anti-Mullerian hormone, but we do have the testosterone being produced, we are going to develop the male, we're going to masculinize, develop penis scrotum, but we're not going to defeminize. So we'll also probably start developing some female genitalia as well. So this is another way in which we could have intersex individuals. All right. So testosterone, estrogen, these are going to stimulate the secondary sex characteristics. Secondary sex characteristics don't typically come into play until post-puberty in humans. And so this is, think about puberty. Think about all the things that people go through during puberty. Voice dropping for males, that's testosterone. Development of breasts, development of pubic hair for both sexes. Those are all secondary sex characteristics. The typical physiological shapes we think about, widening of the shoulders for males, development of the hips for females. Those are all secondary sex characteristics. Okay. All right. We have testosterone also in development here, acting on the Wolffian duct to develop. So here you can see this is the indifferentiated kind of gonad. And you can see we have this thing here called the Mullerian duct, and we have the Wolffian duct here. So we all have both at some point in development. Testosterone is going to develop the Wolffian duct. Estrogen will help develop the Mullerian duct. But female external genitalia will be the default. If we don't have testosterone acting, we're going to develop female. If DHT is present, we'll develop male external genitalia. If DHT is not present, female genitalia. Oh, that's a little blurry. I don't know why. So DHT, if that is present, that means we're producing testosterone and the enzymes are present to convert it to DHT. We will develop the scrotum, we will develop the penis. If DHT is not present or cannot act for some reason, cannot bind to receptors, we will develop external female genitalia, labia. So 6 weeks of gestation, both are there. Then we have testosterone, anti-Mullerian hormone acting on the Wolffian duct to transform it into male. So once the embryonic gonads develop as either testes or ovaries, all the subsequent steps of sexual reproduction are controlled by these sex hormones. It may sound like I'm repeating myself a lot, and I am. And that's because we want to just making sure we keep going through this step by step. Females, the default is going to occur. The Mullerian duct will give rise to the oviduct, connects the ovary to the uterus. The Wolffian duct will degenerate because we don't have the anti-Mullerian hormone or the testosterone acting. Okay. So, indifferentiated gonadal ridge, we're either going to develop into a testes or an ovary. Let's follow the male pathway here first. We have the SRY gene on the Y chromosome. We develop into a testes. We're going to create these cells. The Leydig cells are going to be producing the androgens. This is going to masculinize that Wolffian duct. It's also going to masculinize the external genitalia, and we're developing into male. Because testosterone is converted to DHT by an enzyme called 5-alpha reductase. But this is another place, another kind of complication on our flowchart where things can and often do go awry. If they don't have enough 5-alpha reductase, we're not going to convert to DHT. We're not going to masculinize the external genitalia. That's a common step that can be complicated. The Sertoli cells are going to be developing the anti-Mullerian hormone. We're going to regress the Mullerian ducts, defeminize. If we develop into an ovary, not much is going on hormonally speaking. We're not releasing anything in development anyway. So by default, the Wolferian ducts are going to regress. Mullerian ducts will develop, will feminize. External genitalia will also feminize. So there's a reason why all those examples I gave you at the beginning of these athletes, intersex individuals, are female presenting. Female is the default. When things change on this pathway or things don't go perfectly according to a hormonal plan, female will default, especially externally. So when we think about developing the external genitalia in males, here's a 16-week fetus. You can see the development of the scrotum and the penis beginning to occur here. Testosterone is produced. The enzyme 5-alpha reductase is there to convert to DHT. And it's the DHT that is acting to develop the penis and the scrotum. This is about 16 weeks. This is typically a little around when they start doing anatomy scans when women are pregnant. And that's how they can tell, that's what they're looking for when they determine is the fetus male or female. They're just looking at the external genitalia at that point. Okay. All right, here's a nightmare chart for you all, but it's not such a nightmare. We can walk through it pretty simply. Which do you want to do first, female or male?
Female.
Female. All right, female's the default, so it's easy. This is the only time we get to say the female is the easy pathway. Okay, we have the indifferentiated gonad. Default, we're going to produce an ovary. Ovaries are going to produce testosterone, which is converted to estrogen. Okay, the Wolffian duct will regress because there's nothing there to keep it going. The Mullerian duct will continue to develop. It will develop into the uterus, the upper vagina, the oviduct. The estrogen later in life is going to create those female secondary sex characteristics. And our indifferent external genitalia, default is going to be the female external genitalia. Easy peasy. Female's easy. Now let's do the male pathway. Indifferentiated gonad, we have the expression of the SRY gene on the Y chromosome. We're going to develop into testes. Testes are going to lead to the Leydig and Sertoli cells. Let's do Sertoli first. Going to develop the anti-Mullerian duct hormone, which will cause the Mullerian duct to regress. Leydig cells are producing testosterone. Testosterone later in life, secondary sex characteristics, but it will also start masculinizing the Wolffian duct to develop into the seminal vesicles, the epididymis, the vas deferens. Testosterone is converted to DHT by 5-alpha reductase. DHT is going to masculinize the indifferent external genitalia into a penis and a scrotum. So as you follow the flowchart, as you go through different scenarios, just at each step say, is this present or not? If it's not present, default to female from wherever you are. Okay. So if I'm on this male pathway here, but I take out DHT, I remove 5-alpha reductase, what's going to happen? I'm going to have an individual that has the development of testes, that has internal male genitalia, but they do not have external male genitalia. They have feminized external genitalia. This person would probably be born and raised as female because externally, that's how the infant would present. Okay. Here is another way of looking at it. This is our flow chart. What I like about this flow chart is it shows you some of the free floating connections that can occur. This is a really nice pathway. You can see it kind of linearly here. Female pathway is more simple. Default, if at any point on this male pathway, something goes awry, we don't have the receptors, we're not going to produce the particular hormone, just jump right over at that point to the female pathway and go female from there. Okay. How we doing? How we feeling?
Good.
It is insane. So what the general population doesn't understand is male female is not a simple dichotomy. It's not one thing. It is so many steps here. We haven't even gotten to behavioral, the brain yet. We will in just a minute. Okay. So this chart also includes a little bit of that. They're talking about sexual behavior. So female typical behavior in adulthood, male typical behavior in adulthood. And then they're also looking at here pattern of gonadotropin releasing hormone secretion. So this you'll get into more when you start talking about female and male reproductive behavior, but this is from the hypothalamus, so in the brain again. So we have to not only masculinize, feminize the internal genitalia, the external genitalia, but then we have to masculinize or feminize the brain and various parts of the brain to create different patterns of hormonal secretion later in life and behavior later in life as well.
I have a quick question about this
Please.
flowchart. Is the only way that a chromosomal female XX female that develops external genitalia appearing male, is the only way for that to happen if the SRY gene gets transferred on to an X chromosome?
Probably, yeah. There could be cases where the embryo is exposed to high levels of androgens. in
Oh,
development.
yeah, I remember reading that.
Okay, and that could also create changes.
And if the SRY gene does get transferred on to the X chromosome, that's only happening, I guess, inside of
That's
My-
happening, yeah, that's happening at the production of the gamete level.
in a male.
So that would be the crossover event would have to occur during meiosis when you're creating the sperm essentially.
Mhm.
So it would be happening so that it jumped over and it's an X sperm, but it has that gene.
Okay.
Yeah. So yeah, and that's incredibly rare. That type of event happening in humans, incredibly rare.
Has it been documented though?
I believe there has been 1 or 2 documented cases
Yeah.
in the past 100 years. We really kind of figured it out more from rodent studies where we can do a lot more crossing. And
Yeah, that makes sense. Like genetic manipulation.
exactly. In humans, what is more likely to be a factor in terms of what we would consider intersex? And intersex is going to be, we're going to call it for our definitional purposes, basically anything that does not follow this pathway perfectly on either side. Okay. Much more likely in humans is that there is alterations during the development of the external genitalia and in the hormonal levels. All right. Let's go just a little bit further and then we'll take a nice pause so we can make sure we're up to date on the worksheet. Okay. Although we'll continue the worksheet next time as well. So here's what's kind of going on in utero. 6 weeks pregnancy, we have a sexually indifferent fetus. Around 7 to 8 weeks, we've started to develop male, female. By 15 weeks, the whole process should basically be done. Female development, hormonally independent. It's the default, it's just happening. The male development, we develop the testes, we're gonna have all those hormones, antimullerian, testosterone, DHT. So development of male is completely and 100% hormone dependent. That means this window of time between about 6 weeks to 15 weeks in utero is a crucial time point of development where fetuses are incredibly susceptible to hormonal disruption. If hormones are disrupted in that time period, we're going to start defaulting female and we're going to lead to some intersex characteristics. Right. What's also important to note is this is a time period when a lot of women don't even know they're pregnant yet. So it's a very susceptible time point for disruption, and people aren't even aware of it usually. Okay. So let's talk about a few abnormalities or anomalies really in sexual differentiation. We can have Turner syndrome. This is where someone has XO chromosome. So just one chromosome, one X. They develop abnormal but recognizable ovaries and a feminine body. They do have a reduced IQ. So when we start changing things in the chromosomal level, we can lead to a lot of anomalies. But Turner syndrome, if we don't have that secondary sex chromosome, it'll be female, default female. Here we have Klinefelter syndrome. This is the scenario I kind of brought up earlier, XXY. Because the Y chromosome is there, the gonads will develop as testes and we'll get a more masculine phenotype. Again, we do see reduced IQ, we see sterility, not able to reproduce.
I have a question about Klinefelters.
Sure.
Is there any like indication that it could be like genetic based on how both of the parents, their hormone development? Could it be like that kind of an imbalance in their hormones when the egg is being fertilized? There could be like an issue with that, or is it just based on speculation?
I honestly, I don't know. I'm going to look that up though and get back to you on Wednesday. That's a great question. Dr. Olson, make a note. Okay. He's sitting here listening too. All right. But basically what this kind of underscores is these differences in the chromosomal levels is if a Y chromosome is present, we're still going to develop testes, even if other things are going kind of abnormal. There have been no cases of individuals without an X chromosome at all. Like nobody is YO. That doesn't happen. That indicates that that condition is lethal in early development and will never develop into a adult. Okay. All right. Let's do just one more slide maybe, and then I'll pause. This is a very common syndrome. This is androgen insensitivity syndrome, right? This condition kind of helps us understand the importance of the sex hormones in the development of those secondary sex characteristics. So this syndrome is caused by a genetic mutation. The mutation has to do with the gene that encodes the androgen receptor. The gene is on the X chromosome for this. So males who inherit this condition are producing testosterone, they're producing DHT, they have functional testes that are producing the Sertoli cells, and they're producing all the correct hormones, but they are unable to respond to those hormones. The androgen receptors are not functioning. No receptor, no effect. So these males, chromosomally, are going to develop phenotypically as female. So this is an individual here, this photograph on the left. This is an XY individual who has internal testes, produces testosterone, produces DHT. The body is unable to respond to it. There's a low level of estrogen. Again, remember, I told you the adrenals also produce some of those sex hormones. And the low levels being produced by the adrenals is enough to stimulate the female secondary sex characteristics because female is the default. Okay. Can they have children? Not usually, no. Very likely for most of these scenarios that we're going over, most of the time there's fertility issues as a result. Okay. So again, this kind of underscores two important points for us. One, hormone, basic rule of hormones, no receptor, no effect. And two, the hormones are what are driving these outer development. Okay. All right, I'm gonna pause there. I'm gonna, let's see, stop sharing my screen for a second. And let me see if I can pull up the worksheet too to make sure we're all good on where we should be today. We will finish as we go through. All right. So I'm looking at the worksheet. You again have this in the announcement Dr. Olson sent out. So you want to be able to define the different levels of sex. You got to 18, let's see. Yeah, we just about covered through 18. We'll pick up right around 18 on Wednesday as well to repeat that. We'll also spend a good bit of time on Wednesday working through scenarios. So like on 24 on the worksheet, like we have somebody with this characteristics, what are they going to be male, female wise at the different levels? Okay. So any questions between 1 and 18?
I have a question based on like just kind of a hormonal imbalance in sort of adults. So that the typical one would be like PCOS. Where is that occurring that causes the excess of testosterone? You know, excess facial hair and very masculine like features, and where is that occurring in this whole like kind of flowchart situation? Not in terms of like an embryo developing, but more of as an adult, where is something going wrong?
Yeah, no, absolutely. Those are great questions. You definitely will go through all that when you start talking about female and male reproductive behavior, and that's going to include those types of hormones. But basically what you want to remember is testosterone is produced by both male and female, and testosterone has to be converted to estrogen in female. And there's a hormone or aromatase that does this. So in PCOS, polycystic ovarian syndrome, what happens is basically there's not as much aromatase present. So we're going to have more testosterone present. Does that?
Yeah, that makes sense.
Okay.
What happens if there's like too much estrogen, like that if you become estrogen dominant?
So again, one of my mentors used to like to say, like, life is an inverted U. Basically, too much of anything is going to cause problems.
Of course.
Yeah, so too much estrogen is also going to create certain issues. And basically, things are binding at these receptors, and we think of them as binding exclusively, but it doesn't really happen that way in reality. What really happens is they're more likely to bind at certain places than others. So when you have so much of one of these sex hormones, they might be overflowing and spilling out and tapping into some other receptor systems and creating side effects we would consider negative.
Interesting. Thank you so much.
Of course. Tasha asked if we could go over chromosomal sex, gonadal sex, etc. Absolutely. Chromosomal sex is XX, XY, or whatever those sex chromosomes are. Okay. Gonadal sex is going to be the development of a testy or an ovary. So what does that indifferent gonad develop into? Hormonal sex will be what hormones are being produced more frequently, right? So hormonal sex is going to be everything from testosterone production, anti-Mullerian production during development, but it also includes later in life levels of estrogens testosterone, DHT for those secondary sex characteristics. Morphological sex is going to include both external genitalia. So what kind of think of morphological as phenotypic, like what someone looks like, those secondary sex characteristics. And then behavioral sex, we're going to talk about next time on Wednesday. And that's kind of like programming the brain, so to speak. So male and female typical behaviors specifically related to sex. Okay. All right.
I have another question about the morphological sex.
Sure.
What happens when like a male is developing, or even a female in that aspect, they're developing in puberty and they all of a sudden start having more like female characteristics, or they are developing the normal masculine traits?
So that's actually, typically it's around puberty when most people or their relatives realize that someone might be intersex, because up until that point, they've looked very feminine or masculine or something like that, either way. And then it doesn't, puberty is not triggered. So we're going to start talking about that next time when we think about organizational and activational effects. So like the hormones activating pathways that are kind of laying dormant earlier in life, and how is it going to develop?
Interesting. Thank you.
Of course. All right. Well, it's 10:45, so I will let you all go. I will stick around for just a few more minutes in case you do have any questions, but I'm going to stop the recording now, and Dr. Olson will get that posted for you all.
Thank you so much.
Of course, and I'll see you next time.
Thank you.
I had a